Thank you, Kieran, and good morning to everyone, and thank you to the organisers of the workshop for inviting me to present some of the research that I've conducted as part of my PhD here at UCD. The title of my presentation today is Enhancing Vitamin D Stability and Bioaccessibility Using Mixed Micelles. So the overall aim of my project and the main research goal was to develop a delivery system for vitamin D to both improve the absorption of vitamin D and also to improve the number of products into which vitamin D could be incorporated into. So one of the key challenges we identified at the start was that due to the fat-soluble nature of vitamin D, there's difficulties incorporating vitamin D into non-fat foods. And also that vitamin D is easily degraded when exposed to certain stresses like oxygen, heat, or light. So some of the current strategies for vitamin D delivery include emulsion systems, protein-based systems, and to a lesser extent, mixed micelles. So mixed micelles have had some research conducted in relation to curcumin and vitamin E, so other fat-soluble compounds, but very little research is focused on vitamin D with mixed micelles. So with this in mind, we decided to go down this route of using mixed micelles as a delivery system for vitamin D. So what are mixed micelles? Mixed micelles are colloidal structures formed from fatty acids, phospholipids, and bile salts. On the right-hand side here, you can see a diagram of a mixed micelle. And it's important to note that this is a simplified diagram of a mixed micelle, and that in reality, the structures are far more complex than this. For a reference of the scale of these structures, the mixed micelles I've used in my work are around 4 to 10 nanometers in diameter, so very, very small. When these mixed micelles form, they have a hydrophilic exterior, so they're water soluble, but they have a hydrophobic core in the center, which can host fat-soluble compounds such as vitamin D. So mixed micelles will form naturally in the body during digestion, and they have a key role in the absorption of fat-soluble compounds like vitamin D. So generally, the more mixed micelles that form during digestion and the more vitamin D that's incorporated into these naturally formed mixed micelles will lead to an increase in the absorption of vitamin D. So what we wanted to do then was we wanted to see, could we pre-form these mixed micelles outside of the body, preloaded with vitamin D, with the intention of oral delivery, and that there would be two potential benefits to this. So the first one would be that we may be able to reduce the element of chance of mixed micelles forming naturally during digestion and incorporating vitamin D into their structures. And the second benefit would be that due to the hydrophilic exterior of mixed micelles, they could be incorporated into water-soluble foods or food systems, which may allow for the incorporation of vitamin D into a wider range of food products. So I'm not going to present the initial work that I conducted as part of my PhD involves the optimization of these mixed micelle structures and incorporating vitamin D into the structures. I'm not presenting that work here today, but if you are interested in reading more about them and about that work, I've left a QR code at the end of the final slide, which you can scan with your phone or take a photo of it, and it'll link you straight to that paper, which we published on that. So what I will be focusing on today is the stability of these micelles and the stability of vitamin D within the mixed micelles mostly. So shearing is a common process and stress that happens during food manufacture at several different stages. And high pressure processing is another process that's often carried out on juice and beverage products to improve the shelf life while also maintaining the integrity of the product because it's a non-thermal process. So our aim would be, I suppose, to develop the mixed micelles loaded with vitamin D, and they could be incorporated into a food or a beverage product. So if they're going to be incorporated into another food product, there's a high chance that they're going to experience shearing or high pressure processing at some point during production. So with that in mind, we wanted to see, first of all, would the mixed micelles be stable to shearing and high pressure processing? Because there'd be no point in putting the vitamin D into them if they were going to break down very quickly by being sheared or high pressure processed. So what we did was we prepared samples of mixed micelles. We measured the size of the mixed micelles. We then subjected them to shearing and high pressure processing using the conditions you can see on the screen here. And we measured the size afterwards again, and we observed there were no changes in mixed micelle size, and we concluded that the mixed micelles were stable to this treatment. So with this in mind, we chose to then look at the vitamin D stability in mixed micelles. So for this, we prepared mixed micelles loaded with vitamin D, and we also prepared a control solution, which consisted of water with free vitamin D in the water. We then subjected each of these samples to a heat treatment. So we had a range of heat treatments designed to mimic different pasteurization techniques. We used two different light treatments, either UVC or near UV visible light. And then we also looked at storage stability of the samples at -20 and 4 degrees, so freezer and fridge conditions for up to 4 weeks. And what we did here was we measured the vitamin D content before and after storage or exposure to the stress. And we were able to express the vitamin D concentration afterwards or after storage as a percentage of the initial concentration of vitamin D. So in terms of heat stability, so on the y-axis here, we have the vitamin D concentration as a percentage of the starting concentration we had. And on the x-axis, we have the heat treatments that we used. So the micelles are represented by the green bars and the control samples are represented by the blue bars. So what we saw here was that across all heat treatments we tested, the mixed micelles retained significantly more vitamin D than the control solutions at all heat treatments. This effect was particularly prevalent at longer heating times. So when we heat it for 30 minutes here on the left or for 10 minutes here, we observed a greater difference between the retention of vitamin D in micelle and control samples. But however, we still noticed at shorter heating regimes of 15 seconds or half a second, there was still a significant difference between the retention of vitamin D from micelle and control samples. So from this, we can conclude that the mixed micelles are useful for protecting vitamin D from degradation due to heat exposure. So next we looked at light treatment. So on the left-hand side here, we have near UV visible light, and the green bar represents the micelle and the blue bar represents the control samples. So here, the key point from this graph is that after 20 minutes of exposure to this near UV visible light, the micelle samples retained around 80% of their starting concentration of vitamin D, where the control solutions only retained around 50 to 60% of that vitamin D. So we do see a good protective effect here provided by the mixed micelles. Similarly, when we look at UVC light, this is quite an intense UVC light we used. After 30 seconds of treatment, the vitamin D was completely degraded in the control solutions, whereas around 20% still remained in the mixed micelle samples. So again, we can see here that mixed micelles may provide a good protection to vitamin D, which would be exposed to light during storage or maybe during food processing either. Next, we look at the storage stability. So on the left-hand side, we have the samples which are stored at -20 degrees or in freezer conditions. And again, what we saw was that after 4 weeks of storage, the mixed micelle solutions retained around 65 to 70% of their vitamin D concentration that they started with, where the control solutions only had around 15 to 20% remaining. Similarly, in the fridge conditions then, after 1 week, the control solution, all the vitamin D had been completely degraded, whereas in the micelles, we had around 70% and around 35% after 1 and 2 weeks respectively. So again, we can see here that the mixed micelles may be suitable for extending the shelf life of a product containing vitamin D and for protecting vitamin D during storage. Due to time constraints, I can't go further into detail on these stability studies, but again, I've left another QR code on the final slide that if you'd like to take a picture of it at the end or scan it with your phone, you'll be able to get a link to our published paper based on this work. So as I mentioned earlier, mixed micelles form naturally during digestion and they have a key role in the absorption of vitamin D. So the more naturally if more mixed micelles form naturally during digestion and more vitamin D that's incorporated into these naturally formed mixed micelles will lead to generally increased absorption of vitamin D. So next we wanted to look and see how would our mixed micelle systems compare to maybe a standard emulsion system in terms of delivering vitamin D and improving the amount of vitamin D that appears in that mixed micelle, naturally formed mixed micelles. So for this, we set up an in vitro digestion system based on the InfoGest 2.0 protocol, where we had a mouth stage, a gastric stage, and an intestinal stage. And we had 6 different delivery systems here. So we had 2 oil and water emulsions, one with olive oil and one formed with coconut oil, both containing vitamin D. We then had two more emulsion systems, which were the same, but we had added phospholipid to them. We added phospholipid in this case, as we hoped this might increase the production of natural mixed micelles during digestion. And finally, we had 2 mixed micelle systems, one which was an aqueous-based system, which was the same as what we looked at for the stability study, so just mixed micelles in an aqueous solution with vitamin D incorporated in them. And the second one was this aqueous solution of mixed micelles and vitamin D, but instead incorporated into a protein and lactose-based solution. So what we then did was we digested each of these samples, and at the intestinal stage, after the intestinal stage, we had the intestinal fluid. We centrifuged this fluid to give us a clear mixed micelle phase, which should contain the naturally formed mixed micelles, which form during digestion, as well as vitamin D incorporated in them. And then we should also have a digest phase below that. So what we look at here, when we look at the vitamin D concentration on the y-axis here, and we can see the micelle phase is represented by the green bars, whereas the digest phase is represented by the blue bars. And we have the different delivery systems across the bottom with the micelle base, which is the protein lactose base here, and then the standard micelles here on the right-hand side. So the key take home from this graph is that if we look at the green bars, which is that naturally formed micelle phase following digestion, we can see that across all delivery systems, the amount of vitamin D was not statistically significantly different between any of the delivery systems. So from this, we could conclude, or we could imply that the mixed micelle systems are equally as good or no worse, at least, than an emulsion-based system, and that they may be as good as an emulsion-based system for delivering and improving the absorption and certainly the micellization and bioaccessibility of vitamin D. So to conclude, we found good stability to shearing and high pressure processing of mixed micelles, which is useful if they're looking to be incorporated into food products. We found improved retention of vitamin D on exposure to a range of stresses and during cold storage. The mixed micelles may be at least as good as an emulsion system based on the vitamin D levels recorded in naturally formed mixed micelles following in vitro digestion. The mixed micelles, because of their hydrophilic exterior, may be useful for fortifying non-fat foods with vitamin D. And finally, we don't intend these mixed micelles to replace other fortification methods like emulsions or protein-based systems, or even just using fortified milk, but it's more to add more options and to increase the number of products into which vitamin D could be incorporated into. So finally, I'd like to thank my supervisor, Professor Dolores O'Riordan, and Dr. Graham O'Neill for their help with the work, the Department of Agriculture, Food and the Marine for funding the work. And finally, these are my contact details if anyone would like to get in touch with me after the workshop. And these are just the 2 QR codes if you'd like to take a photo of them for the papers. Feel free. Thank you very much.